So what I'm going to talk about today is how we can use genetics and genomics of pine to inform or supply information to forestry and forestry management. And what I'm going to do is I'm going to use 3 examples, and those are going to obviously relate to the mountain pine beetle. So first of all, I'm going to talk about confirming the host jump. In many insect or any sort of pathogen host system, when there's a new species involved and there's a jump to a new species, it's really important to be able to document that right away. And in this situation, it's a bit trickier, so I'm going to talk a little bit about that and how we use genetics to mitigate problems. Second, climate and environment can often play a really important role in how species are distributed across the landscape. So we took a novel approach and applied genetic data to look at the relationship between environment and species distribution. And then finally, I'm going to talk about how adaptation to the landscape can be used and how we can look for this adaptation to the landscape and then apply this to problems in forestry to ensure forests that have the ability to adapt or deal with variation in changing environments. So first of all, lodgepole and jack pine are different. I didn't know if you guys knew that, but they are. So lodgepole came from the west and colonized, and jack pine came from the east and colonized. And even though they are different, they're similar enough to hybridize. And in that hybrid zone, the hybrids are very morphologically diverse. So a hybrid could look like lodgepole, or it could look like jack pine. So if we have these lovely beetles marching into this hybrid zone and getting close to the jack pine, there was no way for anybody to conclusively say, that's a jack pine that's being attacked by the mountain pine beetle. So what we did is we developed genetic markers that we could distinguish lodgepole, jack pine, and their hybrids with really high accuracy. And so by using these markers, we were able to confirm that jack pine was indeed being attacked by the mountain pine beetle. And while people on the ground in Alberta may have already said, well, we knew that, by publishing these results, it's taken this problem that was very regional, like the rest of the country was saying, oh, let BC deal with it, let Alberta deal with it. It became a national problem. The federal government, provincial governments to the east began to pay attention and ask, what are we doing about this? And even some of those governments are now interacting with our research group to try and figure out what their risks are and how they can mitigate those. So this was, you know, maybe a lot of people in Alberta already knew that jack pine was being attacked, but the nation didn't. So this was really important to be able to conclusively say, yes, it's being attacked by the beetle is attacking jack pine. So we developed these markers and we thought, well, we can do more with these markers than just answer this simple question. And so what we wanted to do is we wanted to take a look at whether we could model the species distribution using the genetic ancestry. So what we did is we calculated the amount of jack pine and lodgepole ancestry. So dark means jack pine, so these have very little jack pine in them, and these have lots of jack pine, and the hybrids have a mixture. And by looking at the environment relationship, we can say where we would expect them to be without sampling the region. So this is a modeling exercise I did with Patrick James, who was working on the project, and he's now at the University of Montreal. And what we found was a very strong relationship between species distribution and these environmental factors. So elevation, mean annual precipitation, and summer heat moisture index all had a negative relationship with jack pine probability, whereas drought and extreme minimum temperature had a positive relationship. So you're more likely to find jack pine, low elevation, cold, and dry. And what we could do with this is develop a distribution map. And so what this is a predicted surface. All the points are our actual sampling points that were used to inform this surface. And in black are the confidence intervals. So on this side of the black line is 95% lodgepole pine, and on this side of the red line is 95% jack pine, and then we have this hybrid zone. And one of the neat things about this, and I think we've already heard from Janice and Nadir, that these two species are different in terms of their response to the beetle. Therefore, if we're going to look at spread risk models, we need to take this into account. So this layer can be directly input into a spread risk model. Because, you know, lodgepole it may go quickly through, or it's going to slow down a jack, or it's going to go quickly through jack pine. So this is a really important result that can be a direct input into the model. So I've already said that lodgepole and jack pine are different. They've evolved in different areas. They have a strong relationship with the environment, so that there's likely they have adaptive variation to that environment. Now, if the environment changes, they might be maladapted to where they're living. For instance, in the south here, mountain pine beetle has been around a lot longer. And here, we have naive trees. So these trees, some of them might have some resistance to mountain pine beetle, whereas these trees have never seen it. And so their environment's changed, they're maladapted. So we want to look at this variation across the landscape and see what adaptations there are and whether we can use them in forestry management or in our spread risk modeling. So we need baseline knowledge to figure out how adaptable these trees are, can we use these, and things like drought. Drought's a really important factor. Maybe some trees are better in really dry environments and we can use their seed stock. But first we need to be able to identify these adaptive traits. So what we've done is we wanted to focus on physiological processes involved with defense. So we wounded trees and we exposed them to drought conditions and we collected their information, their genetic data, we got their genes out of there, basically, and we developed markers from that. And we've analyzed a very large set of these markers on lodgepole, jack pine, and hybrids in Alberta and BC. So from this, we need to identify which ones are adaptive. So there's a kind of a three-step process here. So the first step is to identify basic structure. So this population structure will determine how we look at adaptive variation. Secondly, we need to look at markers that are outside of this norm. So we have population structure, which ones aren't fitting in with that population structure. Then once we get that part done, we look at those outliers and we look at what variables they are correlated with. Are they correlated to drought, moisture? Are they correlated to temperature, elevation, disease? So this is the first stage. This is actually hot off the press. You should be very excited I'm showing you this now. I only looked at it on Friday, so this is like literally hot off the press. And so this is what we found for lodgepole pine. We found two different populations. We found a southern one and a northern one. And if any of you are familiar with some of the work that's been done previously on lodgepole pine, there was some work in 2008 done on mitochondrial DNA by Godbout et al. And they found a similar pattern. So this is likely historical, which is really exciting. I know, you look really excited, almost as excited as I am. But this actually is exciting because this structure is historical. So if we find markers that don't fit this structure, they point towards an adaptation. So that's really important that we found this structure so we can make these comparisons. So of course I don't have all of these analyses done because that's the first step. So I haven't gotten to 2 and 3. But what I wanted to do was show you what it might look like and how it can be used. I find visuals are a little bit more useful. So this is actually a marker we've looked at. This is a species discriminating one. And you can see there's quite a bit of variation across the landscape. So what we want to do is we want to look at the spatial variation of these markers. And what it's overlaid is actually the sea transfer zones for Alberta. And you'll notice there are some correlations with the sea transfer zones, but there's also some large areas where the sea transfer zones are that this marker is the same. So this is an example where maybe these data can be used to redefine these sea transfer zones and make them a little bit more flexible for a changing environment. If the environment changes and we find certain trees are better adapted to those changing environments, then we can use that as seed stock in the future. So we have to think about what the trees were dealing with when they were first established, and then think about what they're going to be exposed to in their future. So just to quickly summarize, genetic approaches can and have been used to inform a variety of parameters in forestry management. By looking at the species distribution, we're able to figure out where our spread risks are, depending on if it's lodgepole, hybrid, or jack pine. By looking at the structure of the lodgepole, we can figure out which markers are adaptive. And then we can look at that adaptive variation and hopefully use that in the future, both for looking at spread risk and for maybe making stronger forests. Obviously, this is really far into the future for some of this, but this is the baseline knowledge to get us to that point. And it's nice that we're already doing this and thinking about this, especially with climate change and changing landscapes. So I'm going to flip it over to Felix, who's going to talk about beetle genomics.
Thank you, Kathy. That's a really good entry point, and I'm glad you've made a number of the genetics points that I would have liked to have made. Thanks to Kathy also in part for some of the slides. Oh, that's not what I should be doing. I should be pushing this one. Just to make it clear, we're not talking about beetles, but especially to Jasmine Janes for many of the results I'm going to show. Jasmine, can you just move your hand back there? That's Jasmine. Jasmine's here from Australia, and where her background is in orchid conservation genetics. And, but she's adapted very well to this new system, and she's also finding exciting things. Now, so here's the beetle. And the markers, as you heard from Kathy, there's a lot of very interesting the markers, but we have looked at two broad classes. The microsatellites. Now you saw how big this TRIA project is, so I'm speaking for quite a number of people that in some cases I was only peripherally involved with. But microsatellites here, let's see, yes, here we go. Microsatellites are small repeated regions of DNA, so 2 to 6 base pairs, for example, would be AAT, 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 that are repeated, and it's the number of repeats that gives you a very sensitive, presumably neutral marker, and 16 loci were developed, so 16 markers, so it's small but still useful number, and assayed in thousands of individuals, of which one, and I'll dwell on this in the next couple of slides, actually allows you to tell the sex of the individual. Then SNPs are just single nucleotide differences, and the assumption is, of course, depends on where it's located, the assumption is by and large you have a greater probability of this being adaptive, and you can try to look at other kinds of things, and then once you know where it's located, that if that one nucleotide difference actually means an amino acid difference, that is then translated and gives you a different protein, then you have some basis for assuming adaptation. Now these numbers, the 1537 and markers, 576 individuals, are not coincidences that they're the same as what Kathy showed you. It's because that's how many you can fit into a particular kind of run. And looking at particular gene families, so candidate gene groups. But what I want to do now with these markers is to show you sort of 4 kinds of things that we can derive from them initially already. Oh yes, I need to get used to this system. Yeah. So for beetle sex ratios, one of those microsatellite loci can be used to determine the sex of a larva, which of course buys you time. You don't have to wait for it to become an adult, and more, it may be a lot more, not just time efficient, but cost efficient and so on. And as you heard from Barry, sex ratio is very important for model parameterization, and I think he's going to be telling you more about that. So this trend assessment assumes a 2-to-1 ratio, where more than 2 females to male, it means you predict an eruption. Lower than that, no eruption, which is already very useful information that presumably, if the models work, is going to allow you to do some prediction. Now, unfortunately, small errors in prediction models can have big consequences. And so it's important to do it right and to test these things. And here's some interesting data. Okay, on the screen I can't even see the map, but you can see here is basically BC, Alberta, BC going down here, and the BC-Alberta border. Interesting trend here. Southern BC and central BC would say that there's some sort of eruption likely, because there's way more than 2, or sometimes about 2-to-1, and sometimes way more than 2-to-1 females to males. Whereas at the leading edge, it's not quite so pronounced here or here, by and large. Seems to be less than 2-to-1. So you would say that maybe that leading edge currently is not what one would predict is likely to erupt. And so I'm going to leave that as a teaser because I know it will be dealt with a little bit later. But certainly these were sampled at a time when they were erupting, and so that part makes sense. Okay, that's one of the ways in which you can actually inform possible management later. Now, another way in which you can get at management, I hope, is to use the data generated by the genes to inform how far they're dispersing. This isn't just what the individuals can do, whether it's 24 kilometers or whether they go up to the jet stream and go further or not. This is sort of the realized dispersal. And this diagram here shows that if there's a real barrier and there's some marker, in this case color, and the dispersal does not go across the mountains, well then these will tend to maintain their perhaps even multi-marker integrity here versus here. But if they're mixing, if this realized dispersal gene flow is happening, then you can get some that are a little purpley here and in here, indicating some degree of mixing. So the genes are moving, and this is realized dispersal, not just the last generation, but the last decade or even the last century. It's hard to know exactly what the historical background is, but it's a different measure of dispersal. And this can be explored here, where, for example, if you just look at the SNPs, these 1,500 SNPs, not all of them were worked out, a smaller number worked out, where just the allele frequencies of the base pairs for each population, take those, put them in a great big matrix, do a principal coordinates analysis, and guess what? That first axis, the first principal coordinates axis, which actually took into account 74% of the variation already, had this nice spread here, which was basically a north-south spread with Canmore, Crow's Nest Pass, and so on, up here to the Fort St. John, Fox Creek, and so on, with just a few little outliers here. So if you redo the analysis, Manning Park and Whistler, for the southern ones, were a little different, and that was captured in the second coordinate axis. So this already is giving you some sense of, again, the realized gene flow, how much mixing there is, and the structure of the populations. All right. Now, another way of getting at this is to look at multi-locus genotype combinations for individuals, which are actually averaged here for each of the populations. The populations are listed down here. And actually, I think if you just sort of look at these two colors, the blue versus the green, in some ways that is parallel to red, or red and blue. Here, it sort of, this program allows you to look at the multi-locus combinations and how they tend to fall into separate so-called populations. If you look at all of these populations simultaneously, then there's a clear sort of north-south gradient in the proportions that fall into each of these two. Whereas if you look at only the northern ones or only the southern ones, then there's a lot less structure within each of those. And in fact, the numbers aren't shown here. This structure is a good deal weaker as well, even though the program has forced it into looking at this sort of two categories. Okay, so once again, there is some structure that is fairly geographically determined. All right, now you can take those two categories here, these two, green and blue, which are now shown as blue and very light blue, and the distribution of those two types is found. There's a transition from one to the other along this zone in here, and if you now do an analysis, it's called an isolation by distance analysis. It's fairly clear that with greater geographic distance, and measuring that against genetic distance, there is increasing genetic distance with geographic distance. That may seem obvious, but basically a transect up here from here to here is a greater distance genetically than from here to here. Whereas within each of those, the northern versus southern, the slope of this line is much less, suggesting that there's a great deal of gene flow in here. There's not a great deal of structure. And there are other things you can look at, like heterozygosity and so on. But that's one of the conclusions. In addition to that, there's a clear north-south sort of separation. The other conclusion is, and I haven't shown statistics for this, but there's a great deal of genetic variation here. These things are not going through a lot of bottlenecks, and they're carrying a lot of their genetic variation with them to new places. Now, these are the SNPs, of which many of them are presumed to be a little bit different, perhaps more adaptive. What about neutral variation? There's a paper now it's no longer in review, it's actually now accepted, based on just the microsatellites, showing those microsat 16 or I guess 15 microsat loci are showing a very, very similar sharply defined gradient in here. All right, so dispersal is being informed. What else might be informed? Well, there is the opportunity now, especially with the SNPs, to try to understand something about adaptation, where you have, for example, if you have a cold-tolerant Mr. Freeze kind of beetle in here that only forms 5% of that population, and it's cold-tolerant, you've got very cold temperatures, and very few of the black guys that aren't able to deal with cold temperatures, whereas the blue guy can. Not so many of those make it through. Just even in a single generation, there can be massive selection and an increase in frequency. Now, this is the sort of thing that we would like to be able to detect. And so 13% of the gene markers from those 1,500 SNPs were candidates involved with cold tolerance in one form or another. Just pick things from a GenBank that seem to be related to thermal regulation in some form. And so in analyzing those at 27 locations, the results are still underway. We're still working on that. It's one of the many things that Jasmine is still working on. But here's some intriguing results where here are 3 SNPs that clearly were not sort of evenly distributed across the landscape, that showed a definite geographic pattern in their frequency of the different SNP alleles. And guess what? For one thing, that pattern is very nicely along this line that you've seen also in the pines. You saw that. And of course that got me thinking is that maybe this isn't so much related to cold, but adaptation to the pines themselves. What's interesting here, and you've got to remember this is correlation, not necessarily causation, is that 2 of the 3 loci that you're seeing here have something to do with, well, their thermal hysterase proteins. In other words, possibly cold tolerance in some way. And the other one is a detoxification gene, a cytochrome 450. So here we have finally the opportunity, if this correlation can be shown to be causation in some way, the opportunity to do some much more fine-grained predictive modeling. And with that, then, I want to point out some of these basic conclusions that were, or applications, if you wish, that the mountain pine beetle population genetics, or genomics, which is genetics writ large, that we can do. So, the ability to sex the larvae to better inform the eruption model predictions, demonstrating that quite strong north-south population structure, which has now been shown both in the microsatellites and the SNPs. But within those regions, there's a lot of gene flow, which is itself useful information for the modelers. even if there's no structure in some region, that's actually nicer. Their models can be a little bit simpler. And then, so also now showing the high dispersiveness and the high genetic variation, which can potentially allow for greater adaptation. And then some work in progress on showing signatures of selection that for loci that might conform right now to the north-south structure, but as soon as they get laid on all those other layers, perhaps we can tease out what is causing that structure. Okay, so much for the population genetics parts, and we'll move on to the next one.